Good day everyone, we are the Group 3 and we are pleased to present a report for Vet Physio 413 on Placentation, the endocrinology of gestation and parturition. So in this report, we will discuss the vital physiological and hormonal processes involved in reproduction from the formation of the placenta to the regulation of pregnancy and the mechanisms that lead to childbirth. Placentation. Placentation refers to the formation of a placenta, an organ that connects the developing embryo to the mother's uterus in placental Eutherian mammals. Placenta is a transient organ of pregnancy that provides an interface for metabolic exchange between the mother and the fetus. The placenta is composed of a fetal component derived from the chorion and a material component derived from modifications of the uterine endometrium. Placental or Eutherian mammals. In placental mammals, the conceptus or the developing embryo attaches to the uterus, wherein it forms a temporary but intimate connection that provides essential nutrients and protection throughout development. This process is known as the intrauterine development, wherein it ensures the safety and nourishment of the embryo until it is ready to survive outside the mother's body. The placenta is a transitional endocrine organ that produces hormones responsible for the maintenance of pregnancy, stimulation of maternal mammary gland, and the promotion of fetal growth. The Eutherian mammals or the placental mammals have a reproductive advantage over oviparous animals or the egg-laying species due to the placenta's role in protecting and nourishing the developing fetus. The final prepartum steps of reproduction are the formation of the placenta, acquisition of endocrine functions by the placenta, and the initiation of parturition, which initially leads to birth and the onset of the lactation. Implantation. Implantation refers to the attachment of the placental membranes to the endometrium in most animals. True implantation is a phenomenon in rodents and humans in which the conceptus buries itself into the uterine endometrium, becoming fully submerged and temporarily disappearing beneath the surface. Wherein in most other species, the conceptus does not truly implant, but rather attaches to the endometrial surface and does not disappear from the luminal compartment. Conceptus components are the amnion, the allantois, and the chorion. The role of the chorion is critical because it forms a part of the placenta, which is essential for the embryo's development. It is also responsible for establishing a connection between the mother and the developing fetus. Chorionic villi. The functional unit of fetal placenta is the chorionic villus. These are small finger-like projections on the surface of the chorion. These tiny villi protrude away from the chorion toward the uterine endometrium. The chorionic villi increases the surface area available for this exchange, making it more efficient. Classification of placentas. Placentas are classified based on the distribution of the chorionic villi, the number of tissue layers separating maternal and fetal blood, or the separation of the blood supply. So as you can see here, we have the diffuse placenta of the horse and pig, the cotyledonary placenta of the cow and sheep, the zonary placenta of the cat and dog, and the discoid placenta of the human and the mouse. Classification of placentas based on the distribution of the chorionic villi. Diffuse placenta. Diffuse placentas have a uniform distribution of chorionic villi that cover the surface of the chorion. So in cell, the diffuse placenta of this cell has a velvet-like surface covered with numerous closely spaced chorionic villi distributed evenly across the entire surface of the chorion. The initial attachment of the placenta occurs around day 12 after ovulation and is well established by days 18 to 20. In mares, like the pig, they also have a diffuse placenta, but it features a specialized microzone known as the microcotyledons. They also have these endometrial cups, which is unique to them. These endometrial cups are temporary structures that vary in size and development between days 35 and 60 of pregnancy, wherein they are formed from both tropoblastic and endometrial tissues. The function of these cups is that they produce this equine chorionic gonadotropin or the ECG. After 60 days, these cups are shed into the uterine cavity and cease to function. So in mares, attachment to the endometrium starts around day 24 and becomes well established by days 36 and 38. The zonary placenta. So the zonary placenta have a band-like zone of chorionic villi found in dogs and cats. The zonary placenta consists of 3 zones. The first one is the transfer zone. The second one is the pigmented zone or the paraplacenta, and the third one is the allantochorion zone. So the transfer zone is a prominent region that forms a broad band around the middle of the chorion. So the primary zone is the primary area for the nutrient and gas exchange between the mother and the developing fetus. Pigmented zone or the paraplacenta. So the pigmented zone is located at both ends of the central exchange zone in certain species placentas. This area contains small blood clots and is highly pigmented. It is thought to play a role in transporting iron from the mother to the fetus. The allantochorion zone. The allantochorion zone is a relatively non-vascular zone found at the distal ends of the chorion. This region has less blood supply and is transparent. This zone may be involved in the absorption of materials directly from the uterine lumen. Discoid placentas. Discoid placentas form a regionalized disc that is found in rodents and primates. It is characterized by having 1 or 2 distinct adjacent discs. These discs contain chorionic villi that interface with the endometrium and provide the region for nutrient and metabolic waste exchange. Cotyledonary placenta. So the cotyledonary placenta have numerous discrete button-like structures called cotyledons. The cotyledonary placenta in ruminants such as sheep and cattle has a complex structure and specific functions that facilitate nutrient and waste exchange between the mother and the fetus. The cotyledon is a specialized placental unit derived from trophoblastic tissues, wherein each cotyledon consists of a network of blood vessels and connective tissues, making it vital for nutrient and gas exchange. So in use, the number of cotyledons ranges from 90 to 100, wherein in cows, the number of cotyledons ranges from 70 to 120. The placentome. The placentome or the point of interface is a functional interface where fetal and maternal tissues connect. It consists of the fetal cotyledon and the maternal cotyledon. The fetal cotyledon is a part derived from the chorion. The maternal cotyledon originates from the caruncles, which are specialized areas of the uterine lining that provides attachment sites for fetal cotyledons. At about day 16 in sheep and day 25 in cattle, the chorion initiates attachment to the caruncles of the uterus. Formation of placentomes. During the attachment process, chorionic villi extend into crisp and the caruncular tissues of the uterus. Crisp are small depressions in the caruncles of the uterine lining in ruminants that allow chorionic villi to anchor during placentome formation. The attachment becomes well established by day 30 in ewes and day 40 in cows, ensuring effective nutrient transfer throughout the remainder of the pregnancy. Convex and concave structures. Convex cotyledon. The cotyledon has a convex outward curving shape. The chorion covers the convex cotyledon, and many finger-like villi extend from the chorionic tissue towards the lumen of the uterus, found in animals like cows and giraffe. Concave cotyledon. The cotyledon has a concave inward curving shape. The chorionic tissue pushes inward, creating a concave interface between the chorion and the maternal caruncle. This is seen in animals like sheep and goats or the small ruminants. So with regards to the growth of the cotyledons, throughout gestation, the size of the cotyledons increases significantly. By the end of gestation, cotyledons in cows can measure 5 to 6 cm in diameter. Placental classification based on the number of placental layers that separate the fetal blood from the maternal blood. The name of each placental type is derived by combining a prefix indicating the maternal tissue and a suffix indicating the fetal tissue. Epitheliochorial placenta. Both the endometrial epithelium in the maternal side and the epithelium of the chorionic villi on the fetal side are intact. This is found in species like sows and the mares. So with regards with the epitheliochorial placenta, so the one as we can see on the picture, is the endometrial capillaries, the 2 for the endometrial interstitium, the 3 for the endometrial epithelium, the 4 for the chorionic epithelium, the 5 for the chorionic interstitium, and the 6 for the chorionic capillaries. Ruminants also have an epitheliochorial placenta, but their endometrial epithelium temporarily erodes and regrows, leading to intermittent exposure of the maternal capillaries to the chorionic epithelium. This is called the syndesmochorial placenta. Binucleate giant cells, which involve in the secretion of hormones and proteins, include placental lactogen and pregnancy-specific protein B. These cells also produce hormones like progesterone and estrogen and are important for the function of the ruminant placenta. Endotheliochorial placenta is defined by the complete erosion of the endometrial epithelium and the underlying interstitial tissue. This erosion means that the maternal capillaries or the small blood vessels are directly exposed to the chorionic epithelial cells from the fetal side of the placenta. So this type of placenta is found in dogs and cats. As we can see in the picture, the number 1, which is the endometrial capillaries, the number 2 for the endometrial interstitium, the number 3 for the chorionic epithelium, the number 4 for the chorionic interstitium, and the number 5 for the chorionic capillaries. Hemochorial placenta. Hemochorial placenta represents an even more intimate relationship between maternal and fetal tissues. In this type of placenta, the chorionic epithelium is in direct contact with pools of maternal blood, meaning there are no intervening tissues between the maternal blood and the fetal chorion, as we can see on the picture below. So this type of placenta is found in primates and in rodents. So here in the picture, as you can see here, the RBC for the red blood cell and the number 1 for the chorionic epithelium that is in direct contact with the pools of maternal blood. So the number 2 for the chorionic interstitium and the number 3 for the chorionic capillaries. It's now time for my fellow group members to take over and report on the endocrinology of gestation and parturition.
Gestation. Gestation is a fascinating and complex process that spans from conception to the birth of an organism. It is during this period that the embryo or fetus develops within the mother's uterus. Factors influencing gestation length: species size and metabolic rate. Larger animals generally have longer gestation periods because their offspring need more time to grow and develop inside the womb. They also have lower metabolic rates, meaning their processes of growth and development take longer. Smaller animals, on the other hand, with higher metabolic rates, tend to have shorter gestation periods as their offspring develop more rapidly. Next is environmental conditions. External factors like temperature, food availability, and stress can influence gestation length. In harsh conditions, some species may experience shorter or delayed gestation as a survival mechanism to protect both the mother and offspring. Third is multiple births. In species that give birth to multiple offspring, gestation can be shorter to reduce the strain on the mother's body. Smaller mammals like rodents often have large litters and shorter pregnancies to accommodate frequent reproduction and quicker developmental cycles. Having multiple offspring also means that the fetuses don't grow as large individually, allowing for a shorter gestation period. Marsupials present an interesting case. Marsupials are a group of mammals characterized by giving birth to relatively undeveloped young. Their gestation lasts only about 33 days, after which the underdeveloped young continues to grow in the mother's pouch, a secondary external environment that nurtures further development. Examples of marsupials are kangaroos. The table shown highlights the variation in gestation lengths across species. Endocrine functions of the placenta. The placenta plays a critical role during pregnancy as a transitory endocrine organ. It produces various hormones that influence both maternal and fetal health. These hormones are essential for stimulating ovarian function, The placenta regulates ovarian hormone production, which is essential for maintaining a healthy pregnancy. Maintaining pregnancy. Placental hormones like progesterone are crucial in preventing early labor and ensuring the uterus remains a supportive environment for fetal growth. Influencing fetal growth. The placenta releases growth factors that help the fetus develop at a steady and healthy rate, stimulating mammary function. In preparation for birth, the placenta promotes the development of the mother's mammary glands, ensuring they are ready for lactation post-delivery. Lastly, is assisting in parturition. As pregnancy reaches its term, placental hormones facilitate the onset of labor. Major hormones produced by placenta. This section explores the roles of two important gonadotropins produced by the placenta. These are equine chorionic gonadotropin and human chorionic gonadotropin. These hormones play vital roles in maintaining pregnancy and regulating reproductive functions. First is the equine chorionic gonadotropin, also known as pregnant mare's serum gonadotropin. It is produced by the endometrial cups of the placenta. It is made by a horse placenta but is used in other animals too. What does ECG does? ECG has lutetropic activity that supports pregnancy. It keeps the pregnancy hormone level stable by maintaining the corpus luteum, which produces essential hormones like progesterone, which is vital in maintaining pregnancy. It also promotes the development of supplementary accessory corpus luteums, which occur due to the ovulation stimulated by ECG between days 40 and 70 of pregnancy. Clinical applications. ECG is commonly used in veterinary medicine to induce superovulation, which helps animals like cows and sheep release more eggs for embryo transfer. However, it doesn't work as well in horses for this purpose. The second major hormone is the human chorionic gonadotropin. HCG is produced by trophoblastic cells of the chorion, a membrane that surrounds the embryo. It begins soon after the blastocyst hatches. The main function of HCG is to maintain the corpus luteum, which is responsible for producing progesterone. Progesterone is critical for sustaining the pregnancy during the early stages. HCG can be detected in urine or blood around 8 to 10 days after conception, and its levels peak around 2.5 months into pregnancy. This hormone is commonly used in pregnancy tests because its presence indicates pregnancy. HCG is also used in veterinary medicine to induce ovulation in non-primate females. Production of ECG and endometrial cups. In mares, ECG is produced by the endometrial cups, which are structures that develop in the uterus. The formation and weight of endometrial cups are directly related to ECG production, as shown in Figure 14.6. This figure illustrates how ECG levels correlate with the weight of the endometrial cups. The ECG levels in the weight of endometrial cups starts to rise around 40 days of gestation, peak, and then gradually decline after 120 days. Endometrial cups are seen in a U-shaped structure. This specimen was removed from a mare at 50 days of gestation. Luteal progesterone output during early pregnancy. After ovulation, the primary corpus luteum forms and starts producing progesterone. As seen in the illustration, at around day 40, ECG production starts to rise, stimulating the primary corpus luteum to continue producing high levels of progesterone. Without ECG, progesterone levels would decline, potentially leading to pregnancy loss. As ECG levels rise, it causes the formation of accessory corpus lutea, the additional CLs, between day 40 and 100. This additional corporal lutea help increase and maintain progesterone levels, ensuring the pregnancy continues. Human chorionic gonadotropin and progesterone production during pregnancy. HCG levels peaks at about 2.5 months of gestation and then begins to decline. Initially, progesterone is secreted by the corpus luteum, but as pregnancy progresses, the placenta takes over as the primary source of progesterone, supporting pregnancy until parturition. Additionally, as placental mass increases between months 6 and 9, a slight rise in HCG levels can be observed, indicating ongoing hormonal activity due to the growing placenta. As seen in the illustration, HCG levels rise early in pregnancy to maintain the corpus luteum, which secretes progesterone to sustain early pregnancy. As the placenta grows, it takes over the production of progesterone. The graph included shows the relationship between HCG and progesterone levels throughout gestation. As HCG declines, progesterone levels rise, indicating the shift from ovarian to placental sources. This section focuses on two critical hormones during pregnancy, which are the progesterone and estrogen. Progesterone is vital for early embryonic development, preventing myometrial contractions through what is known as the progesterone block. In species like ewes, mares, and humans, the corpus luteum is essential early in pregnancy but becomes less important as the placenta takes over progesterone production. In other species such as sows and rabbits, the corpus luteum remains critical throughout pregnancy. Estrogen is primarily produced in the latter stages of gestation, signaling the onset of parturition as its levels peak, signaling the onset of labor by promoting uterine contractions and supporting fetal development. Placental lactogen, also known as somatomamotropin, is a hormone secreted by the placenta. It plays a critical role in influencing fetal growth and mammary function. However, its effects vary by species. For examples, in use in humans, the lactogenic effects are more pronounced than the growth-promoting effects. This hormone is secreted by binucleate giant cells, which are responsible for releasing placental lactogen into the maternal circulation. Placental lactogen is known for its dual actions, the lactogenic and somatotropic functions. It plays a significant role during the latter stages of gestation. The lactogenic activity of placental lactogen promotes mammary function in the dam, while the somatotropic activity promotes fetal growth. Relaxin is a glycoprotein hormone crucial for reproductive physiology. Its production varies between species, originating either from the corpus luteum or the placenta. The synthesis of relaxin is triggered by PGF2 alpha, and it serves a vital function during pregnancy. Primarily, relaxin softens the connective tissues of the cervix and enhances the flexibility of pelvic ligaments, facilitating childbirth. Species producing placental relaxin are humans, mares, cats, dogs, pigs, rabbits, and monkeys. In rabbits, relaxin is produced only by the placenta, while in cows, the placenta does not produce relaxin. In most species, both the ovary and placenta produce relaxin. Use in pregnancy detection. Relaxin is used in commercial pregnancy tests, especially in dogs, where it can be detected in the blood around 30 days into pregnancy. This makes it a helpful tool for early and accurate pregnancy detection. Complex cascade of physiologic events prior to parturition. Parturition starts with signals from the fetus. As the fetus grows, space inside the uterus becomes tight, which puts stress on the fetus. This stress activates a hormone cascade in the fetus, starting with the hypothalamo-pituitary adrenal axis. The fetal anterior pituitary releases ACTH, or the adrenal corticotropin hormone, which stimulates the fetal adrenal glands to produce corticoids or stress hormones. These corticoids cause major changes in the mother's body to prepare for birth. One key change is the removal of the progesterone block, which normally prevents the uterus from contracting. Fetal corticoids help convert progesterone into estradiol, a hormone that promotes labor. This happens through a series of enzymes that convert progesterone into different hormones until it becomes estradiol. At the same time, prostaglandin, specifically PGF2 alpha, is produced. This helps the uterus start contracting and reduces the progesterone even more by causing their corpus luteum to break down. As estradiol and prostaglandins rise, the uterus becomes more active, leading to the start of labor. To conclude, this process starts with the fetus and leads to hormonal changes that prepare the mother's body for birth.
The next topic is parturition. Parturition is the act of giving birth to young, which marks the termination of pregnancy and involves various physiological processes. It is the step in the reproductive process that immediately precedes lactation, uterine involution, and return to cyclicity. It is initiated by the fetus and involves a complex cascade of endocrine events that promote myometrial contractions, dilation of the cervix, expulsion of the fetus, and expulsion of extraembryonic membranes. Here are the signs of approaching parturition in animals. Abdomen continues to enlarge, mammary glands continues to enlarge, and then further secrete milky material. Swelling of the vulva, discharge mucus from the vulva, abdominal muscles relax, causing belly to drop and rump to sink in, and also changes in behavior, such as restlessness, frequently lying down and getting up, frequent urination. For biche and sow, they attempt to build elaborate nests. There are 3 stages of parturition. These are the initiation of myometrial contractions or removal of progesterone block, expulsion of the fetus, and expulsion of the fetal membranes. In stage 1, which is the initiation of myometrial contractions or the removal of progesterone block. In domestic animals, an endocrine signal from fetus appears to initiate contractions. It is generally accepted that the fetus essentially becomes stressed so that it begins to secrete ACTH and corticoids. Fetal cortisol promotes the synthesis of 3 enzymes, the 17-alpha hydroxylase, 17-20-lase, and aromatase, which are involved in the conversion of progesterone to estradiol. In addition to converting progesterone to estradiol, fetal corticoids also cause the placenta to synthesize PGF2-alpha. The PGF2-alpha helps abolish the progesterone block. Removal of progesterone block occurs because fetal cortisol promotes the synthesis of the three enzymes that convert progesterone to estradiol. In some species, the hormone relaxin, which is produced by the ovary or placenta, acts to soften the cervical tissues and also allow the pelvic ligament to stretch to aid in parturition. The synthesis of relaxin is stimulated by PGF2 alpha, and these hormones prepares the birth canal by loosening the supportive tissue so that the passage of the fetus can occur with relative ease. This figure shows the conversion of progesterone to estradiol as parturition nears. Progesterone that is high at the placental interface is converted to 17-alpha hydroxyprogesterone by enzyme 17-alpha hydroxylase. Fetal cortisol also triggers 1720 desmolase to convert 17-alpha hydroxyprogesterone to androstenedione. Androstenedione is converted to estrogen by activation of aromatase enzyme. This conversion removes the progesterone block by biometrial activity. This image presents a cascade of physiological events initiated by fetal cortisol during fetal stress, which is crucial for the onset of parturition. The process begins with fetal stress, which causes the release of fetal adrenocorticotropic hormone or the ACTH. ACTH stimulates the adrenal glands to secrete fetal cortisol, and then this cortisol then triggers several key hormonal changes in the mother. Fetal cortisol induces enzymes that convert placental progesterone or P4 to estradiol or the E2. This shift is significant because progesterone maintains pregnancy while estradiol promotes myometrial contractions, which are essential for labor. Additionally, cortisol stimulates the production of prostaglandin F2 alpha or PGF2 alpha, which leads to luteolysis or the breakdown of the corpus luteum, further decreasing progesterone levels and helping initiate labor. PGF2 alpha also enhances uterine contractions. The release of relaxin, which is prompted by the cortisol, helps prepare the body for birth by stretching the pelvic ligaments. Estradiol enhances secretion in the reproductive tract, aiding lubrication for delivery. As uterine contractions increase, cervical stimulation occurs, leading to the release of oxytocin. Oxytocin then intensifies uterine contractions, creating a positive feedback loop that culminates in maximum pressure, facilitating the expulsion of the fetus. As the levels estradiol increases, coupled with elevation levels of PGF2 alpha, the contracting uterus pushes the fetus towards the cervix. Neural signals from the cervix promote oxytocin release. More contractions, more pressure on the cervix, and more oxytocin and positive cascade. As the fetus enters the cervical canal, the first stage of parturition is complete. The image illustrates the neuroendocrine mechanism of oxytocin released in response to pressure on the cervix and subsequent myometrial contractions. As the fetus moves through the birth canal, pressure increases on the cervix, stimulating sensory neurons. These neurons send signals via afferent pathways to the spinal cord and further up to the hypothalamus. Specifically, this pathway terminates the paraventricular nucleus of the hypothalamus. Upon receiving this neural input, the PVN triggers the release of oxytocin from the posterior lobe of the pituitary gland. The oxytocin is then released into the bloodstream and travels to the myometrium, which is the muscular layer of the uterus. Oxytocin stimulates myometrial contractions, which help propel the fetus through the birth canal. This contraction feedback loop ensures progressive labor and intensifies uterine contractions and making it crucial for the effective expulsion of the fetus. Oxytocin administration is commonly used to induce labor or strengthen uterine contractions in animals with delayed or difficult parturition. The next stage is the expulsion of the fetus. Although estradiol and other hormones increase mucus secretions and general lubrication of the cervical canal and vagina, progressively stronger contractions are critical. Passage of parts of the fetus through the cervix into the vagina, along the rupture of one or both water balls, reflexively initiates actual straining or contractions of abdominal muscle. The combination of uterine contraction and abdominal contraction forces the fetus through the birth canal, and expulsion of the fetus is accomplished. The next stage, which is the expulsion of fetal membrane, or the delivery of the placenta, which normally follows the fetus immediately. Degree of disruption varies depending on the type of placentation, but requires that the chorionic villi are removed from the grapes on the maternal side of the placenta. It is believed to be the cause by the active vasoconstriction of the arteries of the villi and important to minimize hemorrhage. This table shows the stages and duration of parturition among various species of animals and also in humans. Extension beyond what is considered to be the normal upper end duration of parturition constitutes dystocia, which refers to a prolonged or difficult birthing process. Difficulties in parturition usually occurs in second stage, which is the expulsion of fetus. Here are the causes of dystocia. First is the fetal size, which is controlled, for example, by both dam and sire. Larger fetuses can exceed the birth canal size, especially in the first-time mothers. Second is the fetal positioning. Abnormal positioning during birth can hinder delivery. About 5% of the cattle births involve abnormal fetal positioning. Lastly is the multiple births. Twins in monococcus species often cause dystocia due to simultaneous presentation, fetal blockage, or uterine fatigue.